Okay, let's unpack this. If you really boil life down to its absolute core, mechanics, the fundamental challenge, the non-negotiable bit, is movement, isn't it?
It really is.
Delivering fresh supplies and getting rid of waste. Every single living cell, right down to the tiny ones, needs that constant flow, oxygen, fuel, regulatory stuff.
It's a huge continuous transport operation, absolutely.
Like a massive internal logistics network.
And that's precisely where the evolutionary story kicks off. For the really simple life forms, like sponges or coelenterates, they sort of cheated, you could say.
Cheated? How so?
Well, they just used the water around them, circulate the external environment right through their body cavities.
Right. Passive diffusion doing a lot of the work.
Exactly. That was enough for simple substance exchange. But you see, as life got bigger, more complex, well, relying on the outside water just wasn't going to cut it anymore.
So necessity being the mother of invention.
Precisely. The evolutionary pressure drove specialization. Higher organisms, they had to internalize that transport function. They moved from just using water to developing these really complex, self-contained fluids. Fluids designed specifically for moving things around quickly.
And that brings us neatly to our deep dive today.
It does. We're using the NCERT material as our guide here. It details these twin transport fluids, blood and lymph, and of course the incredible machinery that drives them, the human circulatory system.
A powerful engine, really.
Incredibly powerful. And it's a closed-loop system, which is key for sustaining large, active bodies like ours.
Okay, so the NCERT material straight away says blood is the most common fluid used for this job in higher organisms. It's not just red liquid. It's actually described as a living, flowing connective tissue.
That's a great way to think about it, a special connective tissue.
Right. So let's start by stripping it down. What are its actual components?
Well, the fascinating thing is thinking of blood as having this matrix. But unlike other connective tissues, its matrix is liquid. That's the plasma.
Okay, plasma, the fluid bit.
Exactly. And it makes up a big chunk, nearly 55% of the total blood volume. If you were to separate it out, you'd see it's sort of straw-colored, a bit sticky, viscous.
And what's in that plasma? Mostly water, I assume?
You got it, 90 to 92% water. But that remaining, 8 to 10% is where all the action is, functionally speaking. We're talking mainly about proteins. They make up about 6 to 8% of the plasma.
Proteins. Okay, so what kind of proteins are we looking at?
These are the real workhorses. The NCERT highlights 3 key players. First, you've got fibrinogen.
Fibrinogen. Sounds important.
Oh, it's crucial. Its job is entirely about clotting, or coagulation as it's technically called. We'll get into the whole cascade mechanism later, but basically without fibrinogen, even a small cut could be, well, catastrophic.
Right, the leak stopper.
Exactly. Then you have the globulins.
Globulins, like antibodies.
Precisely. If fibrinogen is the emergency repair crew, globulins are the security force. They're primarily involved in the body's defense mechanisms. The whole immune response is built around them.
Yeah, you hear about gamma globulins and things like that.
That's right. They include antibodies, crucial for immunity, both natural and, from vaccines or therapies. Understanding them is fundamental to so much of modern medicine.
Okay, fibrinogen for clotting, globulins for defense. What's the third one?
Albumins. They might sound less dramatic, but they're vital. Their main role is maintaining the osmotic balance.
Osmotic balance? You mean the pressure, sort of?
Essentially, yes. Keeping the right balance of fluid pressure between the blood itself and the surrounding tissues. Without albumin, fluids would leak out where they shouldn't.
Got it. Proteins are key. What else is dissolved in that plasma?
Well, beyond the proteins, plasma is like this constant highway. It's carrying dissolved nutrients, things like glucose, amino acids, lipids, all in transit to the cells.
Fuel delivery.
Absolutely.
Yeah.
And critically, it carries electrolytes too. Those small amounts of minerals mentioned in the NCERT, sodium ions, so Na+, calcium ions, that's C, a double plus, chloride ions, Cl-, bicarbonate, the list goes on.
And these tiny ions are vital.
Absolutely vital for everything from nerve signals firing correctly to muscles contracting. Tiny amounts, huge impact.
Okay. And one more definition before we move off plasma. The NCERT mentions serum. What's that?
Right, serum. It's simple, really. If you take plasma and you remove the clotting factors, basically, you take out the fibrinogen that was left behind. It's called serum, often used in diagnostic tests because it won't clot.
Plasma minus clotting factors equals serum. Okay. That covers the 55% liquid part. What about the other 45%? The formed elements.
Right. The formed elements, these are the cells and the cell fragments floating in the plasma. And the first ones we have to talk about, the real icons of blood, are the erythrocytes.
Red blood cells, RBCS.
Exactly. RBCSS. And they are, by far, the most abundant. The NCERT gives the numbers. A healthy adult male has something like 5 million to 5.5 million of these
5 million?
per cubic millimeter of blood. It's a staggering number. Really emphasizes what their main job is.
Which is carrying oxygen, right, because of hemoglobin.
Precisely. Structurally, they're quite interesting, too. They're formed in the red bone marrow, and they have this unique biconcave shape, like a disc dented in on both sides.
Wide shape.
It's all about maximizing surface area for gas exchange. More surface means oxygen can get in and out more efficiently. And here's a really critical adaptation NCERT points out.
Go on.
In most mammals, mature RBCS are devoid of a nucleus. They ditch it
No nucleus. Why?
to make more room. By getting rid of the nucleus, and other organelles too, they can pack in the absolute maximum amount of hemoglobin, HB.
Ah, the oxygen taxi molecule itself.
Exactly. That iron-containing red, complex protein, it's what actually binds and carries the oxygen. The NCERT states a healthy person needs about 12 to 16 grams of hemoglobin per 100 milliliters of blood. That's how crucial it is for getting oxygen around.
So they're basically just tiny bags packed with hemoglobin. How long do they last?
They work incredibly hard, but they don't last forever. Their average lifespan is about 120 days.
4 months. Then what?
Then they get recycled. They're filtered out and destroyed, primarily in the spleen, which is why the spleen sometimes gets called the graveyard of RBCS.
Efficient recycling. Okay, so that's the millions of RBCS. What other formed elements are there?
Next up, we have the leukocytes, or white blood cells.
The immune cells.
The immune cells, indeed. They're quite different from RBCS. For starters, they're colorless because they don't have hemoglobin, and crucially, they do have a nucleus.
And fewer of them.
Much fewer. The NCERT puts the number at around 6,000 to 8,000 per cubic millimeter, so vastly outnumbered by the RBCS. They're also generally short-lived. Some types, very much so. They're like a specialized army, not the general infantry.
Okay, an army. Are there different divisions in this army?
Oh, yes. The NCERT splits them into two main categories based on how they look under a microscope, specifically whether they have granules in their cytoplasm. So you have granulocytes and air granulocytes.
Granulocytes first, then. The ones with granules.
Right. And the most numerous type here, making up 60 to 65% of all WBCS, are the neutrophils.
Neutrophils, what do they do?
They're the frontline soldiers, the first responders. They are phagocytic cells,
Phagocytic
meaning they engulf things. They literally eat invading bacteria, fungi, or just general cellular debris. Absolutely crucial for fighting off infections quickly.
Okay, the cellular Pac-Man. Who else is in the granulocyte group?
Then you have eosinophils. They're less common, maybe 2 to 3%. Their role seems to be more about resisting certain types of infections, particularly parasitic ones, and they're heavily involved in allergic reactions.
Ah, allergies. So if you have hay fever, your eosinophil count might go up.
Very possibly, yes.
Yeah.
And finally, the least abundant of all WBCS, the basophils. Less than 1%, maybe 0.5 to 1%.
Tiny numbers. What's their job?
They secrete important signaling chemicals, things like histamine, serotonin, heparin. These substances are involved in triggering and managing inflammatory reactions.
Inflammation. Okay, so neutrophils, eosinophils, basophils, those are the granulocytes. What about the agranulocytes and no granule?
Correct, no visible granules.
Yeah.
And this group has two main types. First, the monocytes. They make up about 6 to 8%.
And their function?
They're also phagocytic, like neutrophils. In fact, they're even bigger and more powerful eaters. They often arrive a bit later to the scene of an infection or injury and do the major cleanup operations. They can also develop into even more specialized cells called macrophages in the tissues.
The heavy-duty cleanup crew? And the second type of granulocyte?
The remaining 20 to 25% are the lymphocytes, and these are really the brains of the immune operation.
Lymphocytes. We hear a lot about T cells and B cells.
Exactly. Those are the two major types, the B and T forms, that the NCERT mentions. They are responsible for the specific, targeted immune responses. They can recognize specific invaders, mount tailored attacks, and crucially, they create immune memory.
So they remember past infections. That's why vaccines work.
Precisely. Lymphocytes are central to how vaccines train your immune system. They provide that long-term defense strategy.
Okay, so RBCS, WECS, granulocytes and agranulocytes. Is that all the formed elements?
One more crucial component, the platelets, also called thrombocytes.
Platelets. For clotting, right.
Spot on. But interestingly, they aren't actually complete cells. The NCERT explains they are cell fragments. They bud off from very large cells in the bone marrow called megakaryocytes.
Fragments. Huh. So they're small.
Tiny, but numerous. The normal count is somewhere between 1,50,000 and 3,50,000 per cubic millimeter. So way more than WBCS, but still far fewer than RBCS.
150,000 to 350,000, quite a range.
Yes, the range is quite wide, but staying within it is important. Their main job, as you said, is clotting. They release specific factors that initiate the whole coagulation cascade.
So if your platelet count drops too low?
That's serious trouble. It leads to clotting disorders, you bruise easily, bleed for longer, and in severe cases risk excessive blood loss from even minor injuries. Hashtag Alayathi, blood groups and coagulation.
Okay, we know what blood is now, compositionally. But here's where, well, practical application becomes critical. The NCERT makes it absolutely clear. Knowing the components is one thing, but understanding blood groups is vital for safe transfusions. Not all blood is compatible.
Absolutely. This is where those surface markers on the cells become literally life or death factors. The two major systems we need to understand, as detailed in the NCERT, are the ABO grouping and the rhesus, or RH, system.
Let's start with ABO. How does that work?
It's based on, well, quite elegantly simple principles, actually. It depends on the presence or absence of two specific molecules, antigens, on the surface of your red blood cells. These are called antigen A and antigen B.
Okay, so you can have A or B or both or neither.
Exactly that. That defines the four main blood groups. Group A has A antigen, Group B has B antigen, Group AB has both A and B antigens, and Group O has neither A nor B antigen.
Right, but the NCERT also mentions antibodies in the plasma. How do they fit in?
This is the crucial part for transfusions. Your body naturally produces antibodies against the ABO antigens that you don't have on your own cells. It's a built-in defense.
So if I'm group A, I have A antigens on my cells.
Then your plasma will contain anti-B antibodies. Your body's ready to attack any B antigens it encounters.
And if I'm group B?
You have B antigens and anti-A antibodies.
Okay. What about group AB? I have both antigens.
So you can't have either antibody. If you had anti-A or anti-B, your immune system would attack your own blood cells. So group AB plasma has no anti-A or anti-B antibody.
Makes sense. And group O, no antigens on the cells.
Correct. So your plasma has both anti-A and anti-B antibodies.
Ah, okay. So this immediately tells us who can receive blood from whom.
Precisely. Think about group O. Their red cells have no A or B antigens.
Yep.
So if you give group O blood to someone, their antibodies, whether they have anti-A, anti-B, or neither, won't find anything to attack on those donor cells.
So group O can be given to anyone, the universal donor.
That's right. Group O is the universal donor for red blood cells. But remember, a group O person has both anti-A and anti-B antibodies in their plasma, so they can only receive group O blood.
Right. Now, what about group AB? They have both A and B antigens, but no antibodies in their plasma.
Exactly. Since their plasma lacks those antibodies, they can receive red blood cells from Group A, Group B, Group AB, or Group O without their plasma attacking the donated cells.
Making them the universal recipient.
The universal recipient, correct. The NCERT really stresses how vital careful matching is. If you give someone the wrong type, say B blood to an A person, their anti-B antibodies will attack the donated B cells, causing them to clump together, agglutinate, and break down. This is hemolysis, and it's extremely dangerous, potentially fatal.
Okay, ABO system clear. What about the other one, RH grouping?
The rhesus system, or RH factor, it's named after the rhesus monkeys where this particular antigen was first discovered. It's simpler than ABO in a way.
Yes.
It's mostly about one antigen, the RH antigen, often called RHD. You either have this antigen on your red blood cells, in which case you are RH positive.
Which is most people, right?
Yes, the NCERT says about 80% of humans are RH positive, RH plus V.
Yeah.
Or you don't have the antigen, making you RH negative, RHVV.
Okay, so where's the danger here if an RH negative person gets RH positive blood?
Exactly. Unlike the ABO system where antibodies are naturally present, an RH negative person doesn't initially have anti-RH antibodies.
Yeah.
But if they are exposed to RH positive blood, their immune system will recognize the RH antigen as foreign and start producing anti-RH antibodies.
So the first exposure sensitizes them. The real problem is a second exposure.
Correct. The most critical scenario, and the one the NCERT details carefully, is RH incompatibility between an RH negative mother and an RH positive fetus she's carrying.
Ah, okay. How does that play out?
During the first pregnancy with an RH positive baby, the mother's and baby's blood systems are usually kept separate by the placenta. So generally, no major problems during the pregnancy itself.
But during delivery?
That's the risky point. During childbirth, it's quite possible for some of the baby's Rh-positive red blood cells to leak into the mother's Rh-negative circulation.
And her immune system sees these Rh-positive cells.
Sees the Rh antigen as foreign and starts producing those anti-Rh antibodies. She becomes sensitized.
So no harm to the first baby, but now the mother has these antibodies. What happens in a subsequent pregnancy if the next baby is also Rh positive?
That's the danger zone. Those anti-Rh antibodies produced by the mother are typically of a type IgG that can cross the placenta if they cross into the circulation of the second Rh-positive baby.
They attack the baby's red blood cells.
Exactly. They cause destruction of the fetal RBCS. This condition is called erythroblastosis fetalis. It can lead to severe anemia, jaundice due to the breakdown products of hemoglobin, brain damage, and even stillbirth.
That sounds terrible. Can it be prevented?
Thankfully, yes. It's largely preventable now. The NCERT mentions the standard practice, administering an injection of anti-Rh antibodies, like Rojam, to the Rh-negative mother soon after the delivery of her first and any subsequent Rh-positive baby.
Giving her antibodies prevents her making her own. How does that work?
It's clever. The injected antibodies basically mop up any fetal Rh-positive cells that might have entered her circulation before her own immune system gets a chance to mount a full-scale, long-term memory response. It prevents her from becoming permanently sensitized.
A vital preventative measure. Okay, so that covers the complexities of blood types and transfusion reactions. Let's switch gears to how blood stops itself from leaking out in the first place. Coagulation.
Right, the clotting mechanism. It's an essential response to injury to prevent excessive blood loss. When you get a cut, you see that dark reddish-brown scum forming over it after a little while.
Yeah, the clot or scab.
That's the coagulum, or clot. And if you looked at it closely, the NCERT explains it's mainly a mesh, a network of protein threads called fibrins.
Fibrins, like a net.
Exactly like a net. And this fibrin mesh traps damaged form elements, red blood cells, platelets plugging the leak.
So how does this fibrin net form? It can't just be floating around ready-made, surely.
Absolutely not. It has to be formed rapidly, precisely at the site of injury. And it's formed from an inactive precursor protein that is always floating in the plasma, that fibrinogen we mentioned earlier.
Ah, fibrinogen. So something activates it to become fibrin.
Yes. The enzyme responsible for converting inactive fibrinogen into active fibrin threads is called thrombin.
Okay, so thrombin makes fibrin. Where does thrombin come from? Is it always active?
No, good question. Thrombin also exists in the plasma in an inactive form called prothrombin.
Right, so prothrombin needs activating to become thrombin, which then activates fibrinogen to become fibrin. What activates prothrombin?
This activation is the key step, and it requires an enzyme complex called thrombokinase, also known sometimes as prothrombinase.
Thrombokinase. And how does that get formed? This sounds like a chain reaction.
It is. The NCERT describes it as a cascade process, a series of linked enzymatic reactions. The formation of thrombokinase is triggered by factors released at the site of injury.
What kind of factors?
Well, the injured tissue itself releases certain factors, and, very importantly, the platelets, which aggregate at the injury site and start breaking down, also release crucial platelet factors. All these interact through a complex series of steps to ultimately form the thrombokinase complex.
So injury tissue platelet factors, thrombokinase, converts prothrombin to thrombin, converts fibrinogen to fibrin mesh trap cells, clots forms. Phew.
That's the core cascade. It's designed for rapid amplification. A small initial trigger leads to a large fibrin clot very quickly.
And the NCERT mentions one more absolute critical ingredient for this whole process.
Yes, calcium ions. C++. They play a vital role in several steps of this clotting cascade.
Right.
Without sufficient calcium ions, blood simply will not clot properly. It's indispensable. #tag Eustrauss. Lymph, tissue fluid, and circulatory pathways.
Okay, we've gone deep into blood, its components, types, and how it clots, but blood stays mostly inside vessels. What about the fluid that's actually bathing all our cells, the fluid between the blood vessels and the tissues?
Ah, yes, that's the interstitial fluid or tissue fluid, and it's directly related to blood. The NCERT explains its formation really well. Think about the tiniest blood vessels, the capillaries.
Oh, really thin-walled ones.
Exactly. As blood flows through these capillaries under pressure, some of the fluid component leaks out. Water, small solutes, nutrients, oxygen, they move out through the capillary walls into the spaces between the cells.
What stays behind in the capillary?
Mostly the larger things. The plasma proteins, like albumin, are generally too big to get out easily. And most of the formed elements, the red blood cells, white blood cells, platelets, stay inside the vessel too.
So the fluid that leaks out is kind of like filtered plasma.
That's a great way to put it. Interstitial fluid is essentially plasma minus most of the proteins in cells. Interestingly, the NCERT points out that the concentration of small minerals and ions in this tissue fluid is very similar to that in plasma.
And this interstitial fluid is where the real exchange happens.
Absolutely. It's the crucial middleman. Nutrients and oxygen pass from the blood through the interstitial fluid into the cells. And waste products like carbon dioxide move from the cells through the interstitial fluid back into the blood to be carried away. All exchange happens via this fluid.
Okay, but if fluid is constantly leaking out of the capillaries, doesn't it just build up in the tissues? We'd swell up, wouldn't we?
We would if there wasn't a drainage system. And that's where the lymphatic system comes in. It's this whole separate, elaborate network of vessels running throughout the body.
Like a parallel plumbing system.
Kind of, yes. Its job is to collect this excess interstitial fluid from the tissue spaces. Once the fluid enters these lymphatic vessels, it's called lymph.
Lymph. Okay, so interstitial fluid becomes lymph once it's inside a lymphatic vessel. What's lymph like?
It's generally colorless because it doesn't have red blood cells. Its composition is similar to interstitial fluid, but it also contains specialized cells, particularly lymphocytes.
Ah, the immune cells again?
Yes, the lymphatic system is a major highway for immune cells patrolling the body. Lymph nodes, which are scattered along the lymphatic vessels, act as filtering stations where immune responses can be initiated.
So drainage and immune surveillance. Does lymph transport anything else?
It does. It's an important carrier for some nutrients and hormones. And very specifically, the NCERT highlights its role in fat absorption.
Fats? How so?
Most digested fats are packaged into larger particles in the intestine that are too big to easily enter the blood capillaries. Instead, they're absorbed into special lymphatic vessels in the intestinal lining called lacteals. The lymph then transports these fats eventually into the bloodstream.
Fascinating. Okay, so we have blood in a closed system and lymph in another system draining the tissues. Let's zoom out again and look at the big picture of circulatory pathways across different animals, as the NCERT does.
Right, the evolutionary perspective. Broadly, there are two main types of circulatory systems found in the animal kingdom: open and closed.
Open system, what does that mean?
In an open system, which you find in animals like arthropods, insects, crustaceans, and most mollusks, the blood, or hemolymph, as it's often called, isn't always contained within vessels.
So where does it go?
The heart pumps it through some large vessels, but then it empties out into open spaces or cavities within the body, called sinuses. The hemolymph directly bathes the organs and tissues within these sinuses before eventually finding its way back to the heart.
Sounds a bit less organized.
It is less efficient in terms of directing flow and maintaining high pressure. It works for smaller, less active animals, but for larger, more active ones, you need the closed circulatory system.
Which is what we have. And the NCERT mentions annelids, like earthworms, and all chordates have this.
Correct. In a closed system, the blood is always contained within a continuous network of vessels, arteries, arterioles, capillaries, venules, veins, and never normally flows out into open body cavities.
And the big advantage?
Precision and efficiency. The NCERT clearly states that the flow of fluid can be much more precisely regulated in a closed system. You can control blood pressure, flow rate, and direct blood specifically to the organs that need it most at any given time, essential for complex animals.
Okay. And this evolution towards a closed system is mirrored in the evolution of the heart itself, right? From simpler to more complex chambers.
Absolutely. The NCERT traces this beautifully. Fishes have the simplest vertebrate heart, just two chambers.
Two chambers. Which ones?
One atrium, which receives the deoxygenated blood returning from the body, and one ventricle, which pumps it out.
Where does the ventricle pump it?
It pumps the deoxygenated blood directly to the gills. In the gills, the blood picks up oxygen and gets rid of carbon dioxide. Then, interestingly, this newly oxygenated blood doesn't go back to the heart immediately.
No. Where then?
It flows directly from the gills out to the rest of the body tissues. After supplying the tissues, the now deoxygenated blood returns to the atrium of the heart, completing the circuit.
So the blood only passes through the heart once in each full circuit?
Exactly. That's why it's called single circulation. Simple, but effective enough for fish.
Okay. What's the next step up evolutionarily? Amphibians and reptiles.
Yes, amphibians and most reptiles, the NCERT notes crocodiles are an exception, evolved a three-chambered heart.
Three chambers, so 2 atria and 1 ventricle.
Precisely. They have a right atrium that receives deoxygenated blood from the body, and a left atrium that receives oxygenated blood from the lungs, or skin in some amphibians.
Okay, separate collection, but then...
Both atria empty into that single ventricle. So inside the ventricle, there's some mixing of the oxygenated and deoxygenated blood before it gets pumped out to the body and lungs.
Mixing. That sounds less efficient than keeping them separate.
It is less efficient. That's why this arrangement is called incomplete double circulation. The blood goes through the heart twice per circuit, systemic and pulmonary loops, but the separation isn't complete.
Right. Which brings us to the top tier, mammals, birds, and crocodiles.
On crocodiles, yes. They, along with birds and mammals, have achieved the four-chambered heart, 2 atria and 2 completely separate ventricles.
2 atria, 2 ventricles, fully divided.
Fully divided by septa. This allows for complete separation of oxygenated and deoxygenated blood within the heart. The right side handles only deoxygenated blood to the lungs, and the left side handles only oxygenated blood to the body.
And this is double circulation, complete double circulation.
Exactly. 2 distinct circuits, pulmonary and systemic, with no mixing. It's the most efficient system, allowing for the high metabolic rates needed by birds and mammals.
Okay, let's really zoom in now on our own pump, the human four-chambered heart. The NCERT describes it as a mesodermally derived organ location.
It's located in the thoracic cavity, basically in the chest, nestled between the 2 lungs. It's not perfectly central. It's tilted slightly so that the apex, the pointy bottom part, points towards the left. Thighs-wise, it's roughly the size of your clenched fist.
And it's protected.
Yes, it sits inside a protective sac, a double-walled membranous bag called the pericardium.
Double-walled. Is there anything between the walls?
There is a thin layer of pericardial fluid. This acts as a lubricant, allowing the heart to beat constantly billions of times in a lifetime without generating friction against the surrounding tissues. Reduces wear and tear.
Clever design. Okay, inside. We know 4 chambers. Let's name them properly.
Right. You have the 2 upper chambers, which are relatively smaller and receive blood. These are the atria, the right atrium and the left atrium.
And the lower chambers.
Those are the 2 larger, much more muscular chambers that pump blood out. These are the ventricles, the right ventricle and the left ventricle.
And these chambers are separated.
Yes, by muscular walls called septa. A thin wall, the interatrial septum, separates the right and left atria. A much thicker wall, the interventricular septum, separates the right and left ventricles.
Why is the ventricular one so much thicker?
Because the ventricles have to generate much higher pressure to pump blood out to the lungs or the whole body, compared to the atria just pushing blood into the ventricles next door.
Makes sense. Is there also a separation between the atrium and ventricle on the same side?
Yes, there is. A layer of tissue called the atrioventricular septum separates the atrium from the ventricle on each side. But importantly, this septum has openings.
Openings to let blood through from atrium to ventricle.
Exactly. And these openings are guarded by valves, crucial structures that ensure blood flows only in one direction from the atrium into the ventricle and not backwards.
Valves. Okay, what are they called? Start on the right side, between the right atrium and right ventricle.
That opening is guarded by the tricuspid valve. It's called tricuspid because it has 3 muscular flaps, or cusps, that come together to close the opening.
3 flaps on the right. What about the left side, between the left atrium and left ventricle?
That's guarded by the bicuspid valve because it has 2 cusps. It's also very commonly referred to as the mitral valve.
Bicuspid or mitral on the left. So tricuspid right and bicuspid mitral left stop blood flowing back into the atria when the ventricles contract.
Precisely. When the ventricles squeeze forcefully, the pressure inside them shoots up, and this pressure pushes the cusps of these AV valves, atrioventricular valves, shut, preventing any backflow into the relaxing atria.
Are there other valves? What about where blood leaves the ventricles?
Yes, there are valves there too, guarding the exits into the major arteries. These are called the semilunar valves because their cusps are kind of half-moon shaped.
Okay, where are they located?
There's one semilunar valve at the opening where the right ventricle pumps blood into the pulmonary artery, going to the lungs. And there's another semilunar valve where the left ventricle pumps blood into the aorta, going to the rest of the body.
And their job?
Their job is to prevent blood from flowing back from the arteries into the ventricles when the ventricles relax after pumping. When ventricular pressure drops, the pressure in the arteries snaps these valves shut.
So 4 valves in total, tricuspid, bicuspid mitral, and the 2 semilunars, all working to keep blood flowing forwards through the heart cycle.
Exactly. Unidirectional flow is key to efficiency.
Now, you mentioned the ventricle walls are thicker, especially the left one. Why the difference between right and left ventricles?
Good point. Both ventricles pump blood out, but they pump it to very different destinations with different resistance. The right ventricle only needs to pump blood next door, essentially, to the lungs through the pulmonary artery. That's a relatively low-pressure circuit.
Okay. And the left ventricle?
The left ventricle has the much tougher job. It has to generate enough pressure to pump oxygenated blood out through the aorta to the entire rest of the body from your head to your toes and overcome the much higher resistance of the systemic circulation.
So it needs much thicker, stronger muscle walls to do that. Form follows function.
Perfectly put. The left ventricular wall is significantly thicker than the right.
Now, how does this muscle coordinate its pumping? It beats rhythmically, constantly. Does it need nerve signals for every beat?
That's the amazing part. No, it doesn't. The NCERT explains that the heart muscle itself is special. Parts of it are modified into what's called nodal tissue. This tissue is auto-excitable.
Auto-excitable, meaning it can generate its own electrical impulses.
Exactly. It doesn't need an external nerve command to initiate a contraction. The heart generates its own rhythm. It's myogenic.
Wow. Where is this auto-excitable tissue?
The primary patch, the one that sets the pace, is called the sinoatrial node, or SAN. It's a small area of specialized tissue located in the upper right corner of the right atrium.
The SAN. And it's special because
Because it has the highest rate of spontaneous depolarization. It can generate electrical action potentials faster than any other part of the heart, typically around 70 to 75 times per minute in a resting adult.
70 to 75 beats per minute. That sounds like a typical resting heart rate.
It is. Because the SAN has the fastest intrinsic rate, it drives the rest of the heart. It initiates the wave of excitation that leads to contraction. That's why the NCERT calls the SAN the pacemaker of the heart.
The pacemaker. Okay, so the SAN fires. What happens then? How does the signal spread?
When the SAN fires, the electrical impulse spreads rapidly across the muscle cells of both atria, causing them to contract almost simultaneously.
Pushing blood into the ventricles.
Right. As the impulse travels across the atria, it reaches another specialized patch of nodal tissue, located in the lower left corner of the right atrium, near the septum between the atria and ventricles. This is the atrioventricular node, or AVN.
The AVN, what does it do? Just pass the signal on?
It does pass the signal on, but crucially, it introduces a slight delay. This delay is important because it ensures the atria have finished contracting and emptying their blood into the ventricles before the ventricles start to contract.
Ah, timing control. Smart. So after the slight delay at the AVN, where does the signal go?
From the AVN, the impulse travels down a specialized bundle of fibers that runs through the septum between the ventricles. This is called the AV bundle, or often the bundle of His.
Bundle of His.
And this bundle then divides into right and left bundle branches, which travel down towards the apex of the heart within the interventricular septum.
Taking the signal towards the bottom of the ventricles.
Exactly. And then these bundle branches give rise to an intricate network of fine fibers that spread outwards and upwards through the muscular walls of both ventricles. These are the Purkinje fibers.
Purkinje fibers. And they deliver the signal directly to the ventricle muscle cells.
Yes. This whole conducting system, AVN, bundle of His, bundle branches, Purkinje fibers, ensures that the electrical impulse is distributed rapidly and synchronously throughout the entire ventricular muscle mass, causing a powerful coordinated contraction that pumps blood effectively out into the arteries.
From SAN fire to full ventricular contraction, all coordinated electrically. Amazing. #tag VIY. Cardiac cycle and output measure.
So we have the electrical signal pathway sorted. Now let's look at the actual sequence of mechanical events that this signal triggers the pumping action itself. This whole sequence of contraction, systole, and relaxation, diastole, is called the cardiac cycle.
And the NCERT says one full cycle takes about 0.8 seconds. That's fast.
It is remarkably fast. 0.8 seconds for the whole thing to repeat. Let's walk through it. It usually starts conceptually with a phase called joint diastole.
Joint diastole. Diastole means relaxation, right? So joint means...
Means all four chambers, both atria and both ventricles, are relaxed.
Okay, everyone's relaxed. What's happening with blood flow then?
During joint diastole, blood is passively flowing from the major veins, vena cavae into the right atrium, pulmonary veins into the left atrium. Because the AV valves, tricuspid and bicuspid, are open during this phase, the blood flows straight through the atria and into the ventricles. The ventricles are passively filling up.
And the valves leading out of the ventricles, the semilunar ones.
They are closed during joint diastole. Pressure in the ventricles is lower than in the arteries, so those valves stay shut.
Okay, passive filling. Then the pacemaker fires.
Right. The SAN generates its action potential. This electrical signal spreads across the atria, causing them to contract. This phase is atrial systole.
Atrial contraction. Does this push a lot more blood into the ventricles?
It gives an extra push, yes. The NCERT estimates this atrial kick increases the filling of the ventricles by about another 30%. It just tops them off nicely before the main pumping action.
Okay, atria contract, topping off the ventricles. Then the signal reaches the ventricles via the AVN and Purkinje fibers.
Exactly. And that triggers ventricular systole, the contraction of the ventricles. Now, this is where the pressure really starts to build up inside the ventricles.
And the first thing that happens as ventricular pressure rises.
The pressure inside the ventricles quickly exceeds the pressure in the atria. This pressure difference forces the AV valves, the tricuspid and bicuspid valves, to snap shut.
Preventing backflow into the atria. Is this when we hear the first heart sound?
Yes, that closure of the AV valves is the primary contributor to the first heart sound, the lub.
Lub. Okay. Ventricles keep contracting, pressure keeps rising. What next?
As the ventricles continue to squeeze, the pressure inside them eventually becomes higher than the pressure in the pulmonary artery on the right and the aorta on the left.
Higher pressure inside than outside?
It means the semilunar valves are forced to open, and blood is forcefully ejected from the ventricles into those major arteries. This is the ejection phase.
Right. Ventricle pumps to the lungs, left ventricle pumps to the body. Okay. After ejection, the ventricles must relax.
They do. Ventricular contraction, systole, is followed by ventricular diastole relaxation. As the ventricles relax, the pressure inside them starts to fall rapidly.
And when the pressure inside falls below the pressure in the arteries?
The back pressure in the aorta and pulmonary arteries snaps the semilunar valves shut. This prevents blood from flowing back into the ventricles from the arteries.
And this closure causes the second heart sound.
Precisely. The closure of the semilunar valves at the beginning of ventricular diastole is the main cause of the second heart sound, the dub.
Dub. Okay. Ventricles are relaxing, semilunar valves are closed. What about the AV valves?
As the ventricular pressure continues to drop during diastole, it eventually falls below the pressure in the atria, which have been passively filling with blood again. This pressure difference causes the AV valves, tricuspid and bicuspid, to open.
And blood starts flowing from atria to ventricles again.
Yes. And the ventricles begin passively filling once more, returning us to the state of joint diastole, ready for the whole 0.8-second cycle to begin again.
A continuous cycle, beautifully coordinated. How do we measure how much blood is actually being pumped?
We use two key metrics, both mentioned in the NCERT. The first is stroke volume.
Stroke volume, what's that?
That's the volume of blood pumped out by each ventricle during one cardiac cycle, so per beat. In a typical healthy adult at rest, this is approximately 70 milliliters. 70 milliliters.
70 mLs per beat from each ventricle. Okay, what's the second metric?
The second one is probably more commonly discussed, cardiac output.
Cardiac output, how is that calculated?
It's simply the stroke volume multiplied by the heart rate, the number of beats per minute.
Stroke volume times heart rate. So if stroke volume is 70 millil and a typical resting heart rate is, say, 72 beats per minute.
Then cardiac output would be 70 millimL times 72 beats per minute, which equals 40 milliliters per minute, or approximately 5 liters per minute.
5 liters? That's roughly the entire blood volume of an average adult, isn't it?
It is. So at rest, your heart circulates your entire blood volume around your body every single minute. It's quite remarkable.
And the NCERT makes a really important point here about this value not being fixed.
Absolutely. The body has a fantastic capacity to alter cardiac output based on needs. Both stroke volume and heart rate can change.
Like during exercise.
Exactly. During exercise, your heart rate goes up, and your stroke volume can also increase. Your heart pumps more forcefully. This means your cardiac output can increase dramatically, maybe to 20 or 25 liters per minute, or even more in highly trained athletes.
Wow. That adaptability is key. You mentioned the heart sounds, lub and dub. Are they just incidental noises?
Not at all. As the NCERT implies, they have significant clinical importance. Listening to the heart sounds with a stethoscope is a fundamental diagnostic technique.
So remind me, lub is?
Lub is the first sound, associated with the closure of the AV valves, tricuspid and bicuspid, at the beginning of ventricular systole.
And dub?
Dub is the second sound, associated with the closure of the semilunar valves at the beginning of ventricular diastole.
And doctors listen for?
They listen for the clarity, the timing, and any extra sounds like murmurs. Murmurs can indicate problems with the valves. Maybe they aren't closing properly, leaking, or opening properly, stenosis. These sounds provide valuable clues about the heart's health. Hashtag tag 7. Electrocardiogram, ECG, and double circulation.
Okay, so listening tells us about the mechanical sounds, but we can also measure the electrical activity that drives it all, right? That's the ECG.
Exactly. The electrocardiogram, or ECG, sometimes EKG, depending on the convention, is a graphical representation of the heart's electrical activity during the cardiac cycle.
How do you get that graph?
You use a machine called an electrocardiograph. For a standard clinical ECG, the NCERT mentions attaching electrodes, or leads, to the patient. Typically, 3 leads are used for a basic reading, one on each wrist and one on the left ankle. These detect the electrical currents generated by the heart muscle spreading through the body.
And the graph it produces has those characteristic bumps and spikes. PQRS T waves?
That's right. Each wave, or complex of waves, corresponds to a specific electrical event in the cardiac cycle.
Okay, let's break them down. What does the first little bump, the P wave, represent?
The P wave represents the electrical excitation or depolarization of the atria. It's the visual trace of that SAN signal spreading across the atrial muscle.
And this electrical event leads directly to?
Atrial contraction or atrial systole.
Got it. P wave equals atrial depolarization, leading to contraction. What about the big spiky bit, the QRS complex?
The QRS complex represents the depolarization of the ventricles. This is a much larger muscle mass, so the electrical signal is stronger, hence the larger deflection on the ECG.
Ventricular depolarization, and this immediately precedes.
Ventricular contraction, or ventricular systole. Systole technically begins just after the Q wave starts.
Okay. P wave for atria, QRS for ventricles firing up. What's the last wave, the T wave?
The T wave represents the return of the ventricles from the excited state back to their normal resting state. This electrical recovery phase is called repolarization.
Ventricular repolarization. And the NCERT says the end of the T wave marks the end of systole.
Correct. Once the ventricles have repolarized, they are electrically relaxed and ready for the next cycle.
So how is this ECG trace actually used by doctors?
Oh, it's incredibly valuable clinically. For a start, you can easily determine the heart rate just by counting the number of QRS complexes occurring within a specific time period on the graph.
Simple enough. But it tells you more than just rate, right?
Much more. By carefully examining the shape, duration, and intervals between these waves, P wave, QRS complex, T wave, doctors can identify a huge range of potential problems. Any deviation from the normal pattern can indicate issues like an enlarged heart chamber, insufficient blood flow, ischemia, heart muscle damage, infarction, or problems with the electrical conduction system itself, arrhythmias. It's a fundamental diagnostic tool.
A window into the heart's electrical health. Okay, let's shift from the pump and its controls to the pipes, the blood vessels, arteries and veins. The NCERT describes their structure.
Yes, despite their different functions, arteries and veins share a basic 3-layered structure.
3 layers. What's the innermost one?
The innermost lining is called the tunica intima. It consists of a very smooth layer of flattened cells called squamous endothelium resting on a basement membrane. Its smoothness is key to minimizing friction as blood flows past.
Okay, smooth inner lining. What's the middle layer?
The middle layer is the tunica media. This is composed mainly of smooth muscle cells and elastic fibers.
Muscle and elastic. So this layer can change the vessel's diameter.
Exactly. The smooth muscle allows the vessel to constrict, vasoconstriction, or relax, vasodilation, which is crucial for regulating blood pressure and controlling blood flow distribution. The elastic fibers allow the vessel, especially arteries, to stretch and recoil with each heartbeat.
Right. And the outermost layer?
That's the tunica externa, also called tunica adventitia. It's an outer layer of fibrous connective tissue containing collagen fibers. This layer provides structural support and anchors the vessel to surrounding tissues.
3 layers. Intima, media, externa. Does the NCERT mention any differences between arteries and veins in these layers?
Yes, a key difference highlighted is in the tunica media. It's generally much thicker and contains more muscle and elastic fibers in arteries compared to veins.
Thicker middle layer in arteries. Why?
Because arteries have to withstand much higher blood pressure, the pulsatile force directly from the heart's pumping action. They need to be strong and elastic. Veins, on the other hand, operate under much lower pressure, so their walls can be thinner and less muscular. Veins also often have internal valves, especially in the limbs, to prevent backflow of blood against gravity.
Ah, right. Valves and veins. Not usually in arteries. Okay. Now, these vessels form the two major circuits of our double circulation. Let's recap those.
Right, the beauty of the four-chambered heart is these two separate circuits operating simultaneously. First, you have the pulmonary circulation.
Pulmonary means lungs, so this is the lung circuit.
Precisely. It starts with the right ventricle pumping deoxygenated blood into the pulmonary artery. This artery branches and carries the blood to the capillaries surrounding the air sacs, alveoli in the lungs.
Where gas exchange happens.
Yes, blood releases carbon dioxide and picks up fresh oxygen. This newly oxygenated blood then flows into the pulmonary veins.
Then the pulmonary veins carry it.
Back to the left atrium of the heart, completing the pulmonary circuit. Short loop, vital function.
Okay. Circuit 1, right ventricle, lungs, left atrium. What's circuit 2?
Circuit 2 is the much larger systemic circulation. This supplies the rest of the body.
Starting from
The powerhouse, the left ventricle. It pumps oxygenated blood out under high pressure into the largest artery, the aorta.
the aorta. And that branches out.
Massively. The aorta branches into smaller arteries, which branch into even smaller arterioles, eventually leading to vast networks of capillaries that permeate virtually every tissue and organ in the body.
Delivering oxygen and nutrients everywhere.
Everywhere. At the capillaries, the blood gives up its oxygen and nutrients to the cells and picks up carbon dioxide and other metabolic waste products.
So the blood becomes deoxygenated again. Where does it go then?
This deoxygenated blood collects in tiny venules, which merge to form larger veins. These veins ultimately drain into the two largest veins, the superior and inferior vena cava.
And the vena cava empty into?
The right atrium of the heart, which completes the systemic circuit.
Left ventricle, body tissues, right atrium. So the two circuits run in parallel, ensuring oxygenated blood goes to the body and deoxygenated blood goes to the lungs without mixing.
Exactly. Double circulation. Highly efficient delivery and removal system.
Now, the NCERT also mentions a couple of specialized sort of subcirculations. One is the hepatic portal system.
Ah, yes, the hepatic portal system is unique. Portal means it connects two capillary beds in series. In this case, blood flows from the capillaries in the digestive organs, stomach, intestines, not directly back to the heart, but first goes via a special vein, the hepatic portal vein, to another set of capillaries, sinusoids, within the liver.
So blood from the gut goes straight to the liver before joining the main circulation. Why?
It's a crucial processing step. The liver can then directly process the nutrients absorbed from digestion, like glucose, amino acids, store some of them, and also, importantly, detoxify potentially harmful substances absorbed from the gut before they reach the rest of the body.
A filtering and processing station. Clever. What's the other special circulation mentioned?
The coronary system, or coronary circulation. This isn't about supplying the body or lungs. It's the network of blood vessels that supply the heart muscle itself.
The heart needs its own blood supply. It can't just take oxygen from the blood passing through its chambers.
No, the heart wall is too thick, and the blood passes through too quickly for significant diffusion. The cardiac muscle is working incredibly hard nonstop, so it needs a constant, dedicated supply of oxygen and nutrients via its own coronary arteries, which branch off the aorta right near the start, and coronary veins collect the waste.
So any blockage in these coronary arteries is obviously very serious.
Extremely serious. That's what leads to angina and heart attacks, as we'll discuss. Hashtag tag 8, regulation and circulatory disorders.
Okay, we have the structure, the cycle, the plumbing. But how is it all controlled? We know the SAN sets the basic rhythm, making the heart myogenic, as the NCERT says. But the rate isn't always 72 beats per minute, is it?
No, absolutely not.
While the heart is intrinsically regulated by the nodal tissue, auto-regulated, its activity is constantly being moderated or adjusted to meet the body's changing demands. This modulation comes primarily from the nervous system and hormones.
A nervous system. Which part?
Specifically, a special neural center located in the medulla oblongata of the brainstem oversees cardiac activity. It exerts its influence via the autonomic nervous system, or ANS, the part of the nervous system that controls involuntary functions.
Autonomic, meaning automatic, basically.
Yes, you don't consciously control it. The ANS has two main branches that usually have opposing effects on the heart.
Which are?
The sympathetic nerves and the parasympathetic nerves.
Sympathetic. That's the fight-or-flight system.
That's the one. When the sympathetic nervous system is activated, like during stress, excitement, or exercise, it releases neurotransmitters that act on the heart too. Increase the heart rate, make the SAN fire faster, increase the speed of conduction through the AVN, and increase the force or strength of ventricular contraction.
So overall effect is increased cardiac output. More blood pumped, faster. Gets the body ready for action.
Okay. And the other branch, parasympathetic.
The parasympathetic system is more associated with rest and digest functions. Its nerves, primarily the vagus nerve to the heart, release a different neurotransmitter, acetylcholine, that has the opposite effects.
Slows things down.
Yes, it decreases the heart rate, slows down the SAN, slows the conduction of the action potential through the AVN,
Yeah.
and generally reduces the strength of contraction, though its effect on ventricular contractility is less pronounced than the sympathetic systems. The net effect is a decrease in cardiac output, conserving energy when the body is at rest.
So, a constant balance between sympathetic speeding up and parasympathetic slowing down, fine-tuning the heart rate and output moment by moment.
Exactly. And there's hormonal influence, too. The NCERT mentions that hormones from the adrenal and medulla adrenal and noradrenaline released during stress also increase cardiac output, reinforcing the sympathetic effects.
A complex interplay of controls. Now, what happens when this finely tuned system or the structures themselves go wrong? Let's talk about disorders of the circulatory system. The NCERT covers several important ones, starting with high blood pressure.
Yes, hypertension. Blood pressure, as we know, has two numbers. The NCERT gives the normal benchmark as 120 over 80, 20 millimeter Hg. 120 is the systolic pressure, peak pressure during ventricular contraction, and 80 is the diastolic pressure, minimum pressure when the heart is relaxing.
So what counts as high?
Hypertension is typically diagnosed if a person's blood pressure is consistently measured at 140 over 90 or higher on repeated occasions.
And why is that bad?
Sustained high blood pressure puts extra strain on the heart, forcing it to work harder. It also damages the walls of arteries over time, making them stiffer and narrower. This can lead to serious complications like heart disease, stroke, kidney failure, and damage to the eyes. It's often called the silent killer because it might not have obvious symptoms initially.
Okay, hypertension. Next on the NCERT list is coronary artery disease, CAD, often called atherosclerosis.
That's right. CAD, or atherosclerosis, specifically affects the coronary arteries, the vessels supplying blood to the heart muscle itself.
What happens in these arteries?
It's caused by the gradual buildup of deposits inside the artery walls. These deposits, often called plaques, are made up of things like calcium, fat, especially cholesterol, and fibrous tissue debris.
And these plaques?
They progressively narrow the lumen, the open channel of the artery. This restricts blood flow to the heart muscle.
Less blood flow means less oxygen for the heart muscle. What's the consequence?
If the narrowing becomes significant enough that the heart muscle doesn't get enough oxygen, especially during exertion when its demands increase, it causes chest pain or discomfort. This symptom is called angina, or angina pectoris.
With angina, so it's a pain signaling oxygen shortage to the heart.
Exactly. The NCERT describes it as acute chest pain, indicating insufficient oxygen reaching the heart muscle. It's often triggered by physical activity or stress and relieved by rest. It's a major warning sign of underlying CAD, particularly common among middle-aged and elderly individuals.
A warning sign. Okay. What about heart failure? How is that different?
Heart failure is a broader condition. It's defined as a state where the heart is simply unable to pump blood effectively enough to meet the metabolic needs of the body. The pump is failing to keep up with demand.
So it's not necessarily a sudden event.
Not usually. It's often a chronic condition that develops over time, perhaps due to damage from a past heart attack, long-term hypertension, valve problems, or CAD itself weakening the heart muscle.
The NCERT mentions it's sometimes called congestive heart failure. Why congestive?
Because one of the common consequences of the heart failing to pump efficiently is a backup of blood in the system. If the left ventricle is weak, blood can back up into the pulmonary circulation, leading to fluid congestion in the lungs, causing shortness of breath. If the right ventricle fails, blood can back up in the systemic veins, causing swelling in the legs and abdomen, hence congestive.
Okay, so heart failure is the pump not keeping up. It's important, as you noted earlier, to distinguish this from a couple of other terms people often confuse.
Absolutely, it's crucial. Heart failure is not the same as cardiac arrest.
Cardiac arrest being.
Cardiac arrest is when the heart suddenly stops beating altogether, usually due to an electrical malfunction, like a severe arrhythmia. This causes the person to lose consciousness immediately, and it's a dire medical emergency requiring immediate CPR and defibrillation.
Heart stopped beating, okay? And heart failure is also not the same as a heart attack.
Correct. A heart attack, or myocardial infarction, MI, happens when the blood supply to a part of the heart muscle is suddenly and completely blocked, usually by a clot forming in a narrowed coronary artery, often due to ruptured plaque in CAD. This causes the heart muscle tissue supplied by that artery to die due to lack of oxygen.
So heart attack, muscle damage due to sudden blockage. Cardiac arrest, heart stops beating, often electrical. Heart failure, pump inadequacy over time.
That's a great summary of the distinctions. Understanding these differences is really important. Hashtag tag tag Norex. Conclusion and final thought.
Right, so we've covered a huge amount of ground in this deep dive, really following the NCERT's detailed map. We've looked at the fundamental differences between blood and lymph, the intricate components of blood itself from plasma proteins to platelets.
We've traced the elegant evolution from single to double circulation, dissected the 4 chambers and valves of the human heart, and understood the electrical marvel of the SAN pacemaker driving the cardiac cycle.
And we've seen how that cycle is measured by stroke volume and cardiac output, visualized electrically by the ECG, and how disruptions lead to serious disorders like hypertension, CAD, and heart failure, carefully distinguishing those terms.
You, the listener, should now really have that full conceptual understanding provided by the NCERT. It's like having the complete blueprint for how this essential transport system operates, keeping us alive minute by minute.
From the requirement for C, a double plus ions and clotting, to the meaning of the T wave on an ECG, it's all interconnected.
It truly is. And that precision, especially the regulation we discussed, the ability of the closed system to precisely control blood flow and dramatically increase cardiac output from that baseline, 5 liters a minute, that leads to a final thought.
Go on.
The NCERT highlights how precisely regulated flow is an advantage of the closed system. Think about that adaptability ramping up output for sudden physical demands or stress. It's essential for survival. But then consider, what about the impact of chronic low-level nudges to that system?
Nudges
Like persistent background stress, maybe a diet that constantly triggers low-grade inflammation, perhaps chronic sleep deprivation. These things might not cause immediate disease, but they might constantly be activating the sympathetic nervous system just a little, telling the adrenal glands to release a bit more hormone, demanding slightly higher output, slightly higher pressure, day in, day out.
So not acute stress, but a sort of constant low hum of demand on the system.
Exactly. What might be the long-term consequence for the structural integrity, for the sheer longevity of a system designed to beat maybe 72 times a minute on average for decades if it's constantly being asked to operate just slightly above its baseline? Does that subtle, chronic overregulation contribute to the wear and tear that manifests as disease later in life?
A really powerful question to ponder. The hidden cost, perhaps, of our modern lifestyles on this ancient, exquisitely balanced system.
Something to think about.
Indeed. Well, that brings us to the end of this deep dive.
Thank you for joining with us today. See you next time.